that bolus is going to be moving into that oral pharynx, we now have receptors that are going to be found there, stretch receptors and also chemoreceptors that are found in the wall of the oral pharynx. And that's then going to send a stimulus, send impulses, sensory stimuli, to the deglutition center in the brainstem. So we have a reflex center in the brainstem. Last semester when we were talking about our brainstem, we said there's all these different kinds of reflex centers there. There's three supercritical ones, the three vital ones. What are they? The three vital centers in your brainstem, in your medulla. Cardiovascular center, respiratory center. Yeah, the cardiovascular center is actually two separate centers that sometimes we talk about together as the cardiovascular. So there's the heart, and then there's the blood vessels, right? So that changing the diameter of our blood vessels, changing that cardiac output. So those are absolutely vital centers, absolutely. But then we listed a whole bunch of others as well. There's the sneezing center, and the tickle center, and all these other centers for cough center and swallowing. And so one of those other centers that are, they're not like critical in keeping you alive, but you know, overall they are pretty important. They are going to be stimulated, this deglutition center. And what is that stimulus then going to do? Well, that stimulus is then going to make some changes happen in the structure of your pharynx. And it's going to cause two things to happen. First of all, that soft palate and that uvula that we were talking about last day. What's the uvula? That little kind of dangly thing that's hanging down, exactly right. That's the uvula. And then the soft palate, all that soft tissue, when you put your tongue to the roof of your mouth and you work your tongue posteriorly, and you feel that kind of soft tissue there posteriorly, that's our soft palate. So all of this tissue in here is going to essentially push back against the posterior wall of the oral cavity, or of the oral pharynx. The second thing that's going to happen is that the vocal cords are going to close, so they're going to pull tight across that glottis. And then the epiglottis is going to fold down over the top and make sure that you have a nice seal so that food is not going to be able to go down into the trachea. So once again, there's the epiglottis, that little flap of tissue that we talked about last day. We saw that video of swallowing as well. And so that's going to then fold down and cover over this opening. So now, the only place that the bolus has to go, there is only one pathway that it can take, and that's of course now to go down into the esophagus. So this is when things are working well, and when you're not laughing in the middle of this whole process, which then moves all of that tissue, and then food and drink can get up into the nasal pharynx or the nasal cavity, or it can work its way down into the trachea. So as long as we're breathing normally and we're eating calmly and quietly, and this process works very nicely. On our drawing, we can see the same thing happening. There's that uvula and soft palate blocking access up into the nasopharynx, and then there's our epiglottis that is folded down to make sure that we're not allowing any of that bolus to go down into the trachea. So the only place then that bolus has to go is down into the esophagus. All right, so that's the involuntary phase. That's the second phase of swallowing. But now that we've got that bolus into the esophagus, we now have to get that bolus down the length of the esophagus and into the stomach itself. And so that's our third phase of deglutition, which is the esophageal phase. So in the esophageal phase, we have the upper esophageal sphincter, which is typically going to be closed, is going to relax, and that's going to allow that bolus to enter into the esophagus itself. And that happens again as you are swallowing, and as all of those movements are taking place with the epiglottis and the soft palate, then we're going to have the relaxation of that upper esophageal sphincter. So now the bolus is going to be in the esophagus itself. Initially, as part of your voluntary swallowing process, you're going to be contracting some of the skeletal muscle in the walls of the esophagus to help get that bolus moving down there. But very quickly, as we will see, we transition from skeletal muscle in the superior portions of the esophagus to smooth muscle. And so now we're not going to be relying on ourselves anymore, trying to voluntarily move that bolus in that inferior direction. Instead, we're going to be relying on peristalsis. What did we say peristalsis was? Contraction contractions of the smooth muscle walls of the digestive system. And so peristalsis are these wave-like contractions of the smooth muscle. So this is why when you are hanging upside down from a tree when you're a kid and you're having a snack, because you think it's fun, you stand on your head and you eat a grape. Is it going to work? Is the food going to be able to get to your stomach? Yeah, it will, because this is an active process. You're contracting the walls of the esophagus to move that bolus down to the stomach. Astronauts floating around in space, we're not relying on gravity to move the food down into our stomach. We're going to allow the contractions of the wall of the esophagus to essentially squeeze it down into our stomach. So as the bolus is going to be working its way down by the contractions of the wall of the esophagus, once it gets close to the distal end of the esophagus, the stretch receptors that are found in the wall of the esophagus signal ahead to the lower esophageal sphincter and causes that lower esophageal sphincter to relax. And that then will allow the bolus to move from the esophagus into the stomach itself. So we typically see that all of the different components of the digestive system are going to be isolated one from another via sphincters. So we talked about sphincters before. What have we said about sphincters? We talked about a precapillary sphincter as an example. Yeah, exactly. So they control the flow of substances moving through hollow pathways, whether that's a precapillary sphincter, which is only a single muscle fiber wrapped around the beginning of a capillary, or here, if you've got multiple muscle fibers acting in a ring-like formation, and that's going to be able to constrict and dilate to allow substances to move past those openings in and out of these structures. So the esophagus is going to be essentially isolated from everything else. It's got an upper esophageal sphincter and a lower esophageal sphincter. We'll see that the stomach is isolated because it's got the lower esophageal sphincter, and there's a pyloric sphincter at the other end, and we've got the small intestine that has the pyloric sphincter, and it has the ileocecal sphincter at the other end of that. So there are sphincters all the way along the entire length that essentially are able to regulate the movement of substances from one part to another all the way along. Normally, this lower esophageal sphincter, which is the boundary between the esophagus and the stomach, is going to be closed. Why do we want to make sure that that lower esophageal sphincter in its resting state is going to be closed? Yeah, exactly, to prevent the movement of substances in the stomach from working their way backwards in that opposite direction, working their way in a superior direction. What are we going to see is a major component of our stomach juices? Acid. Well, we don't want that acidity to be splashing back into our esophagus. We want to keep it contained inside the stomach. So we're going to have that lower esophageal sphincter in its normal state being closed. And we're only going to open it when there's a bolus that's moving past there to allow that bolus access into the stomach. All right, so this is our esophageal phase then of the movement of the bolus from the oral cavity down into the stomach itself. When we look at the esophagus, as we said, this idea of peristalsis, or this kind of rhythmic contraction of the smooth muscle in the walls of these gastrointestinal structures, is what's going to cause movement to happen. At the beginning of this unit, we said there's only two things that we can control when it comes to the digestive system. What are they? Motility, right? So the movement. Well, what are we talking about here? This is what we're talking about. It's the movement, the contraction of the walls of the esophagus or the stomach or the small intestine that's going to allow the contents to be able to move further along that pathway. So that's our peristalsis that's going to be used to move that bolus from the pharynx down to the stomach itself. Now, of course, because we're moving through the thoracic cavity and then entering into the abdominal cavity where the stomach is, we have to pass through the diaphragm, because of course the diaphragm is that big muscle that's we should all know about by now, talking about the respiratory system, separating the thoracic and the abdominal cavity, which means you're going to have a hole through the diaphragm to allow that esophagus to pass through, just like you have a hole that will allow the aorta to pass through, and a hole that will allow the inferior vena cava to pass through. When it comes to the esophagus, this hole is called the esophageal hiatus. So if you go on hiatus, what does that mean? You're taking a pause, you're taking a break, right? Some of your classmates are on hiatus today after the test from yesterday. They took a break from coming to class today. Okay, so same idea, we talk about the esophageal hiatus, it's the diaphragm that is on break, or it's on holiday, or it's not existing in that spot, so that you can have an open space so that the esophagus is able to pass through there. If we take a look at the histology of the esophagus, look at the walls, we see that it's the same 4 layers that we're going to be talking about all the time. So we're always going to have a mucosa, there's always going to be that submucosa, there's going to be our muscularis, and then that adventitia, that serosa, that connective tissue on the outside. The difference here that I want to highlight, we said we're going to highlight any kind of interesting things along the way that are a little bit different from what you might expect, is the muscularis when it comes to the wall of the esophagus. And that's what I was mentioning before about this transition between skeletal muscle in the superior portion of the esophagus down to our smooth muscle in the more inferior portions. This is where we say goodbye to voluntary control over digestive function. You decide when you want to chew. I want to take a chew now. And now I want to chew 3 times fast. And now I want to move the bolus from the right side of my oral cavity to the left side of my oral cavity. All of that is voluntary. I'm going to decide when I swallow, when the deglutition process starts. Fantastic. All voluntarily controlled. But as soon as that bolus is now moving into the esophagus, and we're passing through this transition period of the skeletal muscle down into the smooth muscle, we've now given up that voluntary control. We turn over control of digestion to the autonomic nervous system and to the enteric nervous system, that brain of the gut that we were talking about last semester. So all of the nervous tissue associated with the digestive system and all of the hormones that are going to be responsible for controlling digestion. We don't control that anymore. All of that involuntarily controlled digestion begins here at the distal end of the esophagus, and it exists all the way through the stomach, through the small intestine, through the large intestine, and we don't regain voluntary control until the very last moment, the very last few centimeters of the very distal end of the digestive tract, in that anal canal, where there's going to be an external anal sphincter that is going to be skeletal muscle that is voluntarily controlled. And hopefully you're all voluntarily controlling that right now, and we'll talk more about that later on. But this is now the point where we kind of turn over that control, and we don't regain any kind of voluntary control until the very last moment. All right. So this is the histology of the esophagus. If you want to take a look at the inside of the esophagus, there's actually not a whole lot to see. It's just a tube, right? So yes, there's some transitions in terms of types of muscle in the wall, but other than that, there's really not a ton that's going on in the esophagus. So if we take a look at this nice little video, there's the superior portion of the esophagus. You can see some movement that's happening over here, and we're just working our way more and more inferiorly. Mucous membrane along the inner lining. So see, this is pretty boring. There's not a whole lot going on. And so you reach the very distal end, and what is this? That's our sphincter. That's our lower esophageal sphincter. If we were to go through that sphincter, where would we be? Then we would be in the stomach. Exactly right. So again, there's not a whole lot to see in the esophagus itself, because it really is just that tube. Now this movement that's happening right here, what is that? A lot of people want to say peristalsis, but it's actually not peristalsis. We go back a little bit. But that movement that's happening, what do you notice about that movement? Yeah, it looks more like some kind of regularity, some kind of almost like heartbeat that's happening. Yeah, that's exactly right. And that's what that is. And so because, of course, the esophagus is traveling through the thoracic cavity, it's moving past, or it exists right beside the heart. And so this is the heart that's beating, right? It's kind of pushing as the heart is moving. It's pushing on the outside of the esophagus. And so you can see that heart's beating on the outside there. There's that little bit of a narrowing that you can see right here where the camera is. That's that hiatus, that hole in between. So that now we're down into the abdominal cavity. And we've got Okay, well I think that's the firefighters out there. Their personnel alarms. Anyway, okay. This brings us to the stomach. All right, we're working our way through the digestive. Haven't run into any other accessory organs yet. So far it's only been the salivary glands in the oral cavity. And now we're going to be working our way down into the stomach, where we will start to see some more secretions that are going to be happening. So it's a J-shaped sac, as you can see right here. So this is the superior portion of the stomach. This little connection here is what would be attached right in this area. That would be the esophagus, and so that's how the esophagus is going to essentially drain into the stomach itself. It can hold about 1.5 liters, a little bit less than 1.5 liters worth of volume. And it is sometimes described as being sort of having this sausage shape when it's empty, so relatively thin and kind of skinny, but then really does have the capacity to expand and to be able to kind of stretch as it is filled with more and more fluid and bolus. It is found in that left upper abdominal quadrant that we've talked about before, obviously inferior to the diaphragm, but importantly anterior to the pancreas. So a lot of times when we see drawings, and I'll point this out to you a little bit later, they're going to draw both the pancreas and the stomach kind of all together. And so sometimes you'll try to see, well, wait a minute, what is that structure that arrow is pointing to? Is it the stomach? Is it the pancreas? Because they're located anterior and posterior to each other. And so sometimes the textbook will try to draw both. And so you're seeing them like on top of each other. Well, yeah, because one's anterior, the stomach is going to be anterior to that pancreas. So I'll highlight that for you just to take note of as we go forward. When we look at a couple of anatomical features of the stomach, obviously we find that entering into the stomach is going to be that esophagus that we were spending a few minutes talking about before, and exiting from the stomach, we're seeing on the distal end is the duodenum. So this is the proximal portion of the small intestine, and we'll talk more about the structure of the small intestine and all that a little bit later on. When we look at the different parts of the stomach, it turns out that there are 4 different regions that the stomach is divided into. Some of them are going to be more active in terms of that mechanical digestion than others. So we have the cardia. The cardia is this first region that's located kind of just around that access point of the esophagus. Why do you think we call this the cardia? It's close to the heart, exactly right. So it's reasonably close to the heart, even though we know the heart is in the thoracic cavity and we're now in the abdominal cavity. This is the area that's close to the heart. And in fact, the lower esophageal sphincter, the other name for this is the cardiac sphincter. Okay, so the cardiac sphincter has nothing to do with the heart itself. It's not in the heart. It's in the stomach, and it's this boundary between the esophagus and the stomach. But because it's close to the heart, sometimes they call it the cardiac sphincter. But again, when people hear cardiac sphincter, they think, oh, there's some kind of sphincter in the heart. So it's maybe not the best term to use, and that's why they've kind of transitioned away from that and are starting to now use the lower esophageal sphincter more often. All right, so this is the cardia region of the stomach. Then we have the fundus. So the fundus is this kind of superior area of the heart, or of the stomach, that is going to be pretty inactive. Yes, there's muscle there, of course, in the wall, and yes, there will be some contraction that happens, but relative to the rest of the regions of the stomach, it's pretty inactive. There's not a ton of mixing that's going on. Essentially what the fundus is just some extra space. And so when you're eating that gigantic turkey sandwich or that humongous salad or whatever it is that you're eating, you're not able to process all of it all at once. As we're going to see, the duodenum, this first portion of the small intestine, is really only able to process a couple of milliliters, kind of like 1 to 2, sometimes 3 milliliters of stomach contents at a time. Okay, so that's pretty slow, right? Because we want to make sure that it's fully digested and then fully absorbed as much as we can. So we're not going to be eating that big gigantic sandwich and then having it move right through the stomach and into the intestines. It's going to be moving in slow little bits. Okay? But at that point, if you've got all of this content in your stomach, we don't have room for it all to be in the most active parts of the stomach, which are the distal portions. So some of it is going to have to essentially just be kind of pushed off to the side. Okay, we'll deal with you in a little bit. We're just going to kind of shove you over here for a few minutes while we deal with all the rest of this, get it moving off into the intestines until there's a little bit more space, and now we can kind of squeeze you down and then start to process you. So it's just kind of like an extra little storage area inside the stomach.
So when you feel like super full and you feel like that pressure,
Yeah.
That's
right. When you feel super full, it's like pressing up on your lungs. Yeah, that's because you got all that bolus that's up here in that fundus, kind of like squishing up against the inferior surface of your diaphragm and making it hard to breathe. Absolutely, that's right. And moderation, everything in moderation. Don't need to stretch the stomach too much all at once. Okay, so that's the fundus. Then we have the body of the stomach, which is the largest portion of the stomach. It makes up kind of the biggest chunk, kind of in the middle here. It's relatively active, lots of muscle in the walls, of course. As we're getting a little bit towards the distal end of the stomach, the activity picks up, the contractions, the vigorousness of the mixing and the churning increases. So again, a relatively active area. And then the distalmost portion of the stomach is called the pyloric part. It's an interesting name, pyloric part. It's not the pyloric region, it's the pyloric part. So just to keep that in mind, this is this kind of distalmost area just before the transition from the stomach into the duodenum. This is, as I said, the most active part of the stomach. This is where the most vigorous and, we'll say, violent contractions of the wall of the stomach are going to be taking place. And the more active this pyloric part of the stomach is, the more likely it is that the substances from the stomach are going to be moved further down the line into the small intestine.
So when we have like bacteria, for example, H. pylori, when it's actually causing the floor, like a hole in the stomach area.
Yep.
So that's
An ulcer, yeah.
An ulcer, yeah.
An ulcer. I said ulcer. Yep.
Oh.
Yeah.
So then when we have like issues down there, so that would like severely impact the ability for us to actually digest food.
For sure. So when you talk about the formation of ulcers, we can talk a little bit about this when we get into the lining of the stomach and the lining of the small intestine. Essentially, an ulcer is going to be where the stomach acid is eating its way or digesting through the wall of the stomach because there's not enough protection from the mucous membrane there. And that can be triggered by bacterial infection, or if we drink lots of alcohol, for example, sometimes that can wear away the lining of the stomach and allow the acid to be able to essentially burn its way through the wall of the stomach, and that's what an ulcer is. Now it turns out that everybody talks about stomach ulcers as being kind of like, stomach ulcer, stomach. More commonly, actually, are ulcers in the small intestine. And that's because the lining of the small intestine, we don't have the same protection for the lining of the small intestine in terms of a very thick mucus layer as we do in the stomach. The stomach is designed to have acid in it, and so there's lots of protection there. Whereas the small intestine has less mucus to protect it. And so now if we're starting to see too much acid being produced or too much acid moving from the stomach into the small intestine, that can then start generating an ulcer in the duodenum. And those are actually more common than stomach ulcers, even though stomach ulcers that's what everybody thinks. Both of them are essentially the same thing, it just depends where they're happening. What's heartburn? Yeah, so that's what that reflux is that we were talking about before, where that lower esophageal sphincter is not totally closing. And so you're having a little bit of kind of splashback of the stomach contents into the esophagus. And again, just like the small intestine, the esophagus is not well protected to have acid in it. The stomach has lots of thick layers of mucus that we'll talk about to protect it from that acid, but the esophagus doesn't. It's a mucous membrane, but it's pretty thin, it's pretty watery. And so if you have that stomach acid and contents that are essentially splashing back up into the esophagus, that causes damage and pain because the esophagus is saying, I don't want all this acid, it's not supposed to be here. And so that's where you're feeling kind of that that achiness, that kind of pressure that feels like, am I having a heart attack? No, it's just acid reflux. But that's what that is where the stomach contents is moving essentially a little bit, splashing backwards into the esophagus. All right, so these are the four components then that make up, or four areas that make up the stomach itself. There's that lower esophageal sphincter that we were mentioning before. As I said, all of the components of the GI tract are going to be isolated one from another. Essentially, you've got the esophagus as one component, you've got the stomach as another component that are all isolated through the use of these sphincters. So the stomach is going to be kind of on its own between the lower esophageal sphincter and the pyloric sphincter. So the pyloric sphincter, once again, in its resting state is going to be closed. We're only going to open the pyloric sphincter when we want to move the stomach contents a little bit at a time from the stomach into the small intestine. But at any given moment, if you're looking at the stomach and saying, what's open, what's closed, most of the time, those two sphincters are going to be closed, and the stomach is essentially isolated from the rest of the components of the GI tract, doing its own thing. All right, we have two different curvatures associated with the stomach as well. This is just to help us kind of make sure that we're all talking about the same surface. It's a three-dimensional structure. It's got this kind of J shape to it, but which area of the stomach, which surface of the stomach are you talking about when you're looking at the stomach in 3 dimensions? So they use these two curvatures to kind of give us a bit of a better sense of describing where you are on the surface of the stomach. So this is the lesser curvature of the stomach. We've seen this lesser curvature before. Where did we see this? Some omentum. The lesser omentum, exactly. So the lesser omentum is going to be anchored onto the stomach in this lesser curvature of the stomach. That's why they're both called lesser, because they're smaller, right? Smaller curvature, smaller omentum, and so that's what's going to be anchored onto this area. And then we have this other curve here, which is the greater curvature. Okay, so the greater curvature, again, just so you can kind of figure out where you are on the surface. All right, so quick anatomy of that stomach. Now let's look at the wall. So we look at the wall of the stomach. Again, same 4 layers that we've always been talking about. You've got a mucous membrane layer, a submucosa, then you've got your muscularis, and then you've got your serosa on the outside, a connective tissue layer on the outside. But when it comes to the muscularis layer, again, this is the layer that's a little bit different than what we often see in other places. Most other places in the digestive tract, you're talking about the esophagus, the lower portion of the esophagus. If you're talking about the small intestine, we see that this muscularis layer is going to be composed of 2 layers of smooth muscle. There's a longitudinal layer that runs along the length of the structure, so that as it contracts, it essentially makes the intestines, if you want, shorter, and then it relaxes, and then shorter, and then relaxes. That's going to help work things along, move things through the lumen. And we have a circular organization of smooth muscle fibers that run around the circumference. So when they were running around the circumference, how are they going to contract? It's not running this way, it's not contracting and making the structure shorter. It's going to be, compressing it, kind of like a sphincter, but all the way along. And so as we have this kind of alternating contraction and relaxation of this ring lake of smooth muscle all the way along, then you can imagine that we could squish the contents further along that digestive tract. So 2 layers normally, a longitudinal layer and a circular layer that runs around the circumference. But here in the stomach, we have those 2 plus a third. So the stomach, we want it to be so mechanically active, we want so much churning and mixing to take place, we actually have a 3rd layer of smooth muscle in this muscularis. So there's our normal longitudinal and our circular, and now we have this extra one that is this oblique layer. You can see this oblique layer right here. It kind of is going in between the direction of movement, the direction of contraction of the longitudinal and the circular. Right? So if longitudinal and a circular are going at, essentially working at 90 degrees to each other, one's running along the length, one's running along kind of the middle, if you want. This one in the oblique is kind of in between. And so now, because you've got this extra layer of smooth muscle, this is really going to enhance the stomach's capacity for churning and mixing this bolus around with the secretions that's coming from the wall of the stomach. All right, so just to keep that in mind, this extra layer, this 3rd layer of smooth muscle in the wall of the stomach. That brings us to our gastric juice. So we said that we're going to be mixing, and really we should be saying mixing the bolus with this gastric juice, to use the proper term there. But when we talk about gastric juice, what is it? What is the substance that we're going to be finding inside our stomach? Well, there's going to be a bunch of different things, and a lot of them kind of not super friendly to us if they get outside of the lumen of the stomach. So the stomach wall is going to be able to secrete a number of different things. Of course, there's going to be water. There's always water that's going to be secreted. But very importantly, we're going to be secreting mucus. Lots of mucus. A relatively thick version of mucus. And the layer of mucus that lines the stomach, in some places in the stomach, is as thick as 3 millimeters. And you think, well, like 3 millimeters is a lot. Like that's this much. And you're thinking this much mucus that's lining the inner wall of the stomach. Well, that's hugely important because that mucus is going to protect you and protect the tissues of the wall of the stomach from all the rest of the contents. Because the rest of the contents are pretty nasty. You've got some hydrochloric acid that's going to be secreted through the wall of the stomach. Well, hydrochloric, like that's a very, it's a strong acid when you're talking about acid-base balance. This is a super strong acid that you have in your stomach. pH of around 2. And so very, very acidic. What's our normal blood pH? 7.35 to 7.45. And we know that it's a logarithmic scale, so for every full number that you go, that's 10 times more acidic or 10 times more basic. So now we're talking about a pH of around 2. This is very, very, very acidic. And without this protective layer of mucus, very easily is this acid going to be able to essentially burn its way through the wall of the stomach. So hydrochloric acid. That hydrochloric acid is going to do a couple of very important jobs that we're going to talk about in a few minutes. But we do see that the wall of the stomach produces and secretes these hydrogen ions and chloride ions out into the lumen of the stomach. We're also going to see that there's this pepsinogen compound that is produced by the wall of the stomach. And pepsinogen is an inactive protease. What's a protease? It's an enzyme. What does it do? Breaks down proteins, exactly right. Amylases break down starch. Lipases break down lipids or fats. Proteases break down proteins. But we're producing this pepsinogen, which is an inactive protease. You think, well, why are we secreting an inactive protease? And then if you keep reading here about hydrochloric acid, you see that one of the roles of hydrochloric acid is actually to convert and activate pepsinogen into an active form of a protease that we call pepsin. So why don't we just make the pepsin and just release pepsin? Instead of making a pepsinogen, which is this like inactive protease, then it gets released, then it has to mix around with the hydrochloric acid, then it becomes active. Why don't we just release an active protease?
Well, the entire gland of the stomach technically that would not be protein, so if it was constantly active, eventually it would eat through the actual gastric line.
Exactly right. What's the major constituent of a cell?
Protein.
Protein, right? The major building blocks of cells, one of them, is protein. And so if you are producing, as a stomach lining cell, if you are producing an active protease, what is that protease? As soon as you make it, what is that going to start to do to you as the cell that's making it? Yeah, breaking your own proteins down. And we don't want that. So we release the inactive version. Once it gets to the other side of that protective mucus layer, into the lumen of the stomach, well, now we can activate it, mix it around with the hydrochloric acid, fantastic. Now it's an active protease, and because it's on the other side of the mucus, we don't have to worry about our own stomach lining actually being digested by that protease.
So just to confirm, so pepsinogen, we're secreting it from the wall, correct?
From the wall, exactly.
So structurally, it would make sense for it to not be active for that sake of reason. So once it's
Correct.
in the lumen, then we can
Once it's out in the lumen and kind of on the other side of that protective layer of mucus, now we don't have to worry about it anymore. We can become active, and it's not going to be able to then target the proteins in the wall of the stomach, because again, that mucus is there to protect us.
Just so that I can understand how important that is. So the pepsinogen is produced within the mucus.
Pepsinogen is produced by the cells in the wall of the stomach.
Oh, okay.
Yep.
Okay. And then when it comes out in its active form, that's to prevent it from damaging our own stomach cells.
Yep.
And then once it's inside the actual stomach itself, where things are ready to get digested, then it can go to its active form.
You got it.
All right, great.
Yeah, exactly. Yeah, it becomes active when it comes into contact with the hydrochloric acid in the lumen of the stomach, and of course then you can be an active protease there because we want to digest the protein that's in the food that's coming in, but we don't want it to digest the wall of the stomach itself. Great. Okay. So once we've got all of these secretions, this water and this mucus and carcinogen and hydrochloric acid, all these things that are being produced by cells in the wall of the stomach, together we call all of these secretions gastric juice. Okay, so gastric juice, not something that you would want to drink out of your fridge, but very important secretions that are going to be crucial in digestion. So when we start to talk about the different types of digestion that go on in the stomach, we're going to see that this gastric juice is going to play a very big role, especially in the chemical digestion of our food. As we're going to do in all of the different components of the GI tract, we're going to talk about the two different types of digestion. We talked about the two different types of digestion in the oral cavity, our mechanical digestion and our chemical digestion. And so we're going to do the same thing here in the stomach. So for our mechanical digestion, as we know, there is going to be this bolus that you have now swallowed, enters into the stomach through which sphincter? That lower esophageal sphincter enters into the stomach, and then once it gets into the stomach, it's going to encounter these kind of nice, slowly mixing waves, just welcoming it into the stomach, this bolus that's there. That's great. You've got every 15 to 25 seconds or so in that cardia region of the stomach, there's some contraction. There's going to be some initial mixing together, just introducing the bolus to the gastric juice and saying you're going to become very good friends over the next little while, mixing it together. And then eventually that bolus realizes that it's actually been totally kind of mixed in with all of these gastric secretions. And now we don't call it a bolus anymore. Instead, we call it chyme. And I know that everybody wants to pronounce this chime or chimey or something with that ch sound at the beginning, but it's not. It's chyme. And I did confirm with Aaron last night. I just wanted to make sure that we were both on the same page about how there was no conflict. This is how you pronounce this word. So everything is fine at my house last night. And so we have chyme is the pronunciation for this watery mixture. So now you've started with food. You've mixed it with saliva to form a bolus. Now you take that bolus and you mix it together with our gastric juice, and you now get a much more watery version of this mixture that we call chyme. Okay, so we've got these kind of nice gentle waves, the generation of our chyme that's being produced, and now as the contents of the stomach, again, are kind of being churned and mixed around, eventually it's going to be churned and mixed around, and it's getting a little bit further along in the stomach. It's moving from the cardia region down into the body, so we're starting to see more vigorous waves now of contraction. We've got those 3 layers of smooth muscle in the muscularis, and so there's going to be kind of more intense waves and some more intense contracting that's going to be taking place. And then really by the time we get to that pyloric region, or that pyloric part, there's a lot of contraction and pretty vigorous and violent contractions that are taking place. And that's then going to make sure that we are fully mixed, that there's a lot of that mechanical digestion that's going to be taking place. And then at the very distal end of the stomach, in that pyloric part, we're going to have a little bit of this chyme that is moved out into the duodenum. As I said, only kind of a couple of milliliters at a time is going to be moved from the stomach out into that duodenum, because the duodenum really can't handle a huge volume all at once. And so we want to kind of have this, allow time for the digestion to continue in the duodenum, in that first portion of the small intestine. All right, so this is our mechanical digestion that's taking place. You'll notice that in the fundus, that kind of storage area of the stomach, not a lot going on there, because again, the focus is more towards that distal end, and then as we work our way through the contents in that distal end, in the body and in the pyloric part, well then the contents in the fundus will work its way down and then be processed as well. All right, so we've got this mechanical digestion then that is taking place in the stomach, but of course if we've got enzymes, we talk about proteases, then there's going to be some chemical digestion that's taking place as well. So the chemical digestion, the big one here in the stomach is protein. Okay? There's not going to be a lot of digestion of other types of compounds, especially not carbohydrates. So that salivary amylase that we just a little bit got started in the mouth, that's going to be destroyed by these proteases that are here, because essentially what kind of compound or what kind of molecule is an enzyme? If you had to pick one of our macromolecules, protein, carbohydrate, or fat, which of those three is an enzyme? Protein. So enzymes are proteins. Well, now we're saying that in the stomach we're going to start to digest protein. Well, one of those proteins is actually our salivary amylase that we produced in our saliva. So it's going to be broken down, and it's not going to be able to continue its digestion of carbohydrates. Carbohydrate digestion, we're going to have to wait until we get into the small intestine to pick up that story. But we do see that there's going to be some protein digestion that begins here. And it's going to happen in a couple of different ways, or a couple of different mechanisms to make this happen. First of all, that hydrochloric acid that we're talking about is part of our gastric juice. Yes, it's going to activate that pepsinogen into pepsin, as you can see listed there again, but it's also going to denature the proteins. What does it mean to denature a protein?
Changes shape of configuration.
To what?
Changes shape.
Unravel it. Exactly right. You may have done this morning. Where we run into denaturing proteins pretty regularly in your life. Eggs. Exactly right. So if you cracked an egg this morning, right, you crack that egg open, tons of protein in that egg, and it starts out the egg white is pretty clear, colorless, maybe a little bit yellowish, kind of. And then you let it cook for a bit. What does it do? It becomes all opaque, right? It becomes kind of really dense and really hard. Why? Because you're denaturing it. You're unraveling it. And remember that proteins have three-dimensional structures. Proteins are amino acids, individual amino acids that are all linked together through peptide bonds. And then you take this big long string of amino acids and you fold it up into a three-dimensional shape. And now you have your functional protein. Well, what we can do is unfold them. Like you said, you take that three-dimensional structure and you start unfolding it back into its long kind of string-like configuration of simply amino acids all attached together. That's what denaturing is. You can do that a couple of different ways. You can do it with heat when you're cooking your egg, or you can do it with acid. So if you were to take an egg and crack it into a bowl full of vinegar, what would happen to your egg? Kind of the same thing. You're kind of cooking your egg, but in acid this time rather than with heat. Same idea is taking place where you're denaturing the protein. What that does, what this acid and this denaturing does, is by unfolding those three-dimensional structures of the proteins, it makes it easier for the enzymes to be able to get in, to start chopping up those individual chemical bonds, those peptide bonds in between the amino acids. You can imagine if you've got a big three-dimensional ball, well, the enzymes are only going to be able to attack from the outside and only be able to break down those chemical bonds from the outside. To make it more efficient, you want to unfold that, expose all of those chemical bonds to the enzymes, so now the enzymes can get in and really start to break that down into their subunits. So we're going to get those peptide bonds broken, and then we're going to be able to get those amino acids. Okay, so we've got our protein digestion that's going to begin. We see a little bit of fat digestion that's going to take place, but not a ton, especially in adults. So we do have gastric lipase, but gastric lipase is in a pretty low concentration, and it's not super efficient at breaking down fats. What its major job is actually when we are infants. Because what is our major diet when we are infants? Milk, right? So most of what we're going to be taking in, a large portion of what we're taking in, is going to be milk and milk fats. Whether that's human milk fats or other types of milk fats, it's milk fats. And so we want to be able to digest and give as much opportunity to be able to absorb all of those nutrients as possible as Junior is trying to grow as quickly as possible. So we do have the gastric lipase that is going to be splitting those fats in our milk. But again, in adults, because the percentage of our diet that is actually milk fat is pretty low, we see that it doesn't have a super large role in adults. Okay, as we said, carbohydrate digestion does not happen in the stomach. We'll have to pick up that story when we get to the small intestine. So we're going to take our 10-minute break. When we come back, we're going to talk about how we regulate this gastric function. How do we make